Welcome, everybody, to the SEBS Virtual Science Cafe. Our cafe today is Learning from Chestnut Blight to Control Plant Diseases with Dr. Bradley Hillman. My name is Brian McGonigal. I'm the Manager of Alumni and Community Engagement at SEBS and the New Jersey Agricultural Experiment Station. Just a few notes before we get started. Our event is being recorded, and it will be posted online and shared in a follow-up email to all registrants and attendees, along with Dr. Hillman's contact info. If you have questions for Dr. Hillman, you are free to enter them in the Q&A box at the bottom of your Zoom screen at any time during his presentation. After he is done with his presentation, we will answer them in turn with time permitting. Right now, I'd like to introduce Dr. Mary Nucci. Mary is the Assistant Dean of Community Engagement and also an Assistant Professor in the Department of Human Ecology. She is the Director of the Science Cafe program. Mary, take it away.
Thank you very much, Brian, and welcome everybody to today's Science Cafe. I'd just like to introduce Dr. Hillman to you. Dr. Hillman received his BS, MS, and PhD degrees at the University of California at Berkeley, where he characterized the first defective interfering RNAs for plant virus. As a postdoctoral fellow here in New Jersey at the Roche Institute of Molecular Biology, he initiated studies on viruses associated with reduced virulence and biological control of the chestnut blight fungus, which he has continued to study throughout his academic career. He joined Rutgers in 1989, where besides chestnut blight, his research has focused on viral diseases of blueberry and cranberry, potato late blight pathogen, fungal pathogens of turf grass, and genome characterization of the eastern filbert blight fungus. And from 2006 to 2020, in his spare time, he served as Director for Research and a Senior Associate Director for the New Jersey Agriculture Experiment Station. And we welcome you, Brad, and thank you very much for speaking to us today.
Thanks so much, Senator Nucci, Senator-elect, is that it? And thank you for organizing this. I'm going to try to share my screen now. See how that goes. Okay.
It's come up, Brad, you're good.
Okay, good. Thanks. Thanks again for organizing these. I think that that science communication is just such an important piece of what we do, and frankly, most of us don't do it enough. And so thanks again for this. What I'm going to try to do is go through first a general introduction to chestnut blight, a little bit of breeding, which I don't do, and then talk a little bit about biological control. We work on kind of complicated systems on viruses that are associated with biological control. I'll try not to get into the weeds too much about those, but I do want to introduce them because one of the sort of bottom line messages, I think, of what I do here, what we do is you start out with application-based goals. So we start out looking at these really as biological control vehicles. And in the end, what you want to make sure that you do is do good science along the way, so that, you know, some of those application-based goals will be met and some won't. But if you do good stuff, then you'll learn things that are applicable to other systems as well. So that's one of the overall messages here, I think. I want to point out some information, some resources that are available to folks. One that's been a huge resource for the chestnut community is the founding of the American Chestnut Foundation almost 40 years ago. Anybody who's interested in this should join that organization. They put out a very nice publication, a very nice magazine, but also it's a great community of people. People are really passionate about chestnut. And another resource is a very readable book by Susan Frankel. Came out about 15 years ago or so, It's a little bit dated now, but really not much. It's called American Chestnut, Life, Death, and Rebirth of a Perfect Tree. And so anybody who is interested, take a look at those. We've been associated with a USDA multi-state project that came about. I think it was initiated in 1986, just before I started working in this area. And it's a project that's been going strong ever since then. We won a regional award for the project, and we won a national award for the project a number of years ago. A lot of the folks who are seen here are people who have since retired or left otherwise. So there's a new community of folks associated with the project as well, though the project is now in its, I think, 6th or 7th iteration. It's now NE 1833. So getting into chestnut, chestnut is worldwide in distribution. There are arguably, the taxonomists argue over it, but I think there are now 9 recognized species of chestnut. American chestnut, which we'll talk about the most, is Castanea dentata, and is here in North America. There are also 2, and I think they're now elevating it to 3 chinquapin species that are related to American chestnut. These are all blight susceptible. So when I talk about chestnut blight, all of these are very susceptible to blight. European chestnut is moderately susceptible to chestnut blight, not quite as susceptible as American, but it is definitely susceptible. And then Asian chestnut, Chinese and Japanese chestnut are quite resistant to blight, and so they're used in breeding programs that we'll talk about. So none of these are as stately a tree as American chestnut, and so that's one of the reasons that American chestnut is so beloved. Chestnut came several million years ago from Asia, probably originating in the Urals, and eventually came over probably across the Bering Strait, although that's I think that's controversial, and was largely disappeared during the Ice Age, and then reemerged as ascended as a major hardwood species some years ago, 2,000 years ago. So again, a very stately tree that could dominate forests and could be 20 to 30% of growing areas of forested areas. And that was until chestnut blight arrived in the late 1800s, early 1900s. You can see, and I love this picture of chestnuts in pollination, probably in June, where there's so much pollen on a hill like this that's just covered with chestnut that it looks like snow frosted trees, but in fact that's just all chestnut pollen. So again, chestnut blight came it was first found in 1904 in the Bronx Zoo, the Bronx Botanical Garden, but we're pretty sure that actually arrived into New Jersey probably in the late 1800s, because it was already established, and so a number of people have done work to show its to show that it was probably brought here, probably from on Japanese chestnut that were brought in for breeding purposes. And so by 1950 or so, the blight had wiped out American chestnut all the way through its range, through its natural range and extended range. and this really changed lives of a lot of people. It certainly changed the ecology of these areas. So when I say wiped out, a lot of you have seen pictures like this of the so-called ghost forests. So those forests that I showed earlier on mountainsides. When something like this happens, it just completely alters the ecology and the economics of an area. A lot of folks in these areas, the Appalachians, were dependent on chestnut for their livelihoods at various levels. And this also coincided with a real increase in logging through these areas. So it was kind of a double whammy for the trees and the ecology. Blight is a filamentous ascomycete fungus that is a so-called necrotroph. It can kill trees quickly. So when chestnut blight infects a small tree like this, it can kill it within weeks, certainly within months, and can take down large trees within just a few years. In the natural range now, chestnut continually resprouts, and so we still have a lot of chestnut out there, and some of these trees actually grow to maturity. And of course, what this does is it keeps chestnut coming back. It also keeps chestnut blight. It also keeps the fungus out in the community as well. And so we still have some good sized trees in New Jersey. I'll say a couple of words about them. These are pictures taken in New Jersey and eastern New Jersey, out near Sandy Hook, where we've done a fair amount of collecting and research out there. We started, actually, Peter Bedker, who is a former faculty member here, started collecting samples here a long time ago. And this is a postdoc of mine, Jim Polishok, who is still is now at the Marucci Center working on blueberry and cranberry diseases. And this picture was taken out near the Earl Naval Weapons Base in Monmouth County. We have some stories associated with that that are kind of interesting. We mapped some of the trees in this area a few years ago. We haven't done a lot of mapping and not too much collecting in the areas, but I worked with Rich Leopold, who just got his PhD in ecology and evolution with Jason Grabowski a month or so ago. And he helped us map some of the trees in 3 park areas here, again, in eastern New Jersey, near the Atlantic Highlands area, and 3 parks in Tyndall Park and in Huber Woods and in Hartshorn Park, and estimated based on counts that there are about 5,000 or so trees, American chestnuts, that are bigger than, I think we had a cutoff of 3 inches diameter. But so again, there are a good number of trees still out there that appear to be pure American trees. And there are stories like this all over the country, in Michigan, in Connecticut, in Maine, in other parts of the country. There are communities of trees that are making a comeback. I'm going to talk now a little bit about breeding and efforts to develop and deploy resistance to chestnut blight. The breeding program, again, has been led by the American Chestnut Foundation, first by a guy named Fred Hebard, who is actually a plant pathologist who got into breeding. And the way the breeding program was done, for many, many years, breeding was just not done effectively at all But then Charles Burnham came in and with the idea of doing classical backcross breeding, that is taking a very blight resistant tree such as Chinese chestnut in this case, and crossing it with a very susceptible tree such as in this case American chestnut. And you want the American chestnut characteristics to the tree, and so you cross those trees and then try to cross out the Chinese genes except for the blight resistance genes. And so you cross it and then you back cross the blight resistant progeny to blight susceptible Americans again to try to get more American germplasm into the crosses, so that finally after multiple back crosses you end up with trees that are majority American genetic material and a minority of in this case Chinese genetic material, but what you've contributed from the Chinese tree is the blight resistance. This looked like it was going to work. It looked like there were probably, folks thought at first that there were only 2 or 3 genes associated with blight resistance, and so this seemed like a good strategy. The bottom line is that it has turned out not to be, so they got to the stage of having 15/16, a backcross 3 tree that was 15/16 American and 1/16 Chinese, and that still, so that had decent blight resistance, but not enough. And so those efforts are going on, as I'll mention in a minute. Those trees, the 15/16 tree, American trees, were used by Stephen Handel and Christina Konziger in the Ecology and Evolution department to look at restoration ecology, look at gap filling of spaces at Duke Farms, and this was in collaboration with the American Chestnut Foundation. And so that was an interesting project based on those first generation backcross trees. And so you can certainly communicate with Stephen and Christina if you want to learn more about that interesting project a few years ago. Another approach, and one that has been led by Bill Powell at SUNY in Syracuse, is to make transgenic chestnut. Bill gave a seminar here that some of you may have seen last fall. He's been working on this for over 30 years now. And the idea here was to bring in a single gene, the oxalate oxidase, OXO gene from wheat that confers disease resistance in wheat, and put that wheat gene into chestnut. The chestnut does not have a homologue, a copy of that particular gene. Turned out, he tried a number of genes, turned out that this one worked, and this one made blight resistant trees. And he achieved that about 15 years ago. It was a lot of work to do, and has been a lot of work since. But now they have these trees that are very blight resistant trees and that are looking very good, and that they're in the process of trying to get regulatory permission for release of these trees. So again, if you want to learn more about this, visit Bill's website. He's got a number of good videos associated with it as well. So a very interesting line of work. And what's happening now with the breeding efforts is sort of a combination of those two, of the backcross breeding program and using genomics of the tree. We know the genome structure of Chinese chestnut, and American chestnut is in pretty good draft mode now. So using the things that you can learn from the genomics of the tree to add to the breeding program to ensure also that by using a lot of different genetic materials from American chestnut, that you end up hopefully with genetic diversity and blight resistance in the end, because you don't want a monocrop of a transgenic tree, for example, where they all have the same basic genotype. So the idea is to make sure that you get a lot of different American characteristics in the trees that are still blight resistant. So this effort is now being led by Jared Westbrook of the American Chestnut Foundation, who is a very well-trained breeder. I'm going to switch and now talk about the viruses and hypovirulence and biological control of chestnut blight. This was actually sort of predated the efforts to the real efforts to do breeding, because it looked so promising at first. But first I'm going to say one thing. The disease chestnut blight is a fungal disease. So the fungus infects the tree. I'm going to talk about viruses a little bit. Viruses don't infect the tree. The viruses infect the fungus, and the fungus infects the tree. A lot of people just get this confused early on, so they keep thinking about what is the virus doing to the tree. The answer is the virus is doing nothing to the tree. It's the fungus that's infecting the tree, and the virus is infecting the fungus. And so this was discovered in the early 1950s in Italy, this phenomenon, where recovering trees were found. And when fungus was isolated from some of those recovering trees, this is not an Italian tree, but was the idea, when when fungi were recovered from those recovering trees, they looked different. The fungus looked different than the fungus coming from dead or dying trees. This fungus was orange, grew fast. This fungus was white, grew slowly. And over the years, we found that, and many researchers contributed to this, it was found that this morphology was transmissible from one culture to another and within trees. So in other words, there was something transmissible in the fungus that was making the fungus sick, essentially, and allowing the tree to recover from the fungal infection. And so, in fact, you could, and people still do, take a tree that is being killed, has a canker that is killing the tree, and take a virus containing, a fungus that contains virus, and just put little cork bore holes around that canker with the fungus that is the hypovirulent, or the less virulent form of the fungus, and ask it to transmit its virus from this fungus to the fungus that's killing the tree. And in fact, that often works. So years later, you can look at the same tree, and in fact, it has recovered. The canker hasn't killed the tree like it would have otherwise. What this looks like in culture is the transmissibility of the virus in the fungus looks like this. In this case, the virus containing fungus is not white, it's this light brown color. But the virus infected culture, when it meets a virus-free culture, the virus is actually transmitted. The hyphae actually fuse, and the virus is transmitted across this so-called anastomosis bridge from the virus-containing fungus to the previously virus-free fungus, and it causes the change in the phenotype that you can see here. Whereas if you don't get that fusion, if the two fungi can't fuse together, then the virus-free fungus stays virus-free, and the virus-containing fungus has kept its virus, and it was not able to transmit. This so-called vegetative incompatibility here or compatibility here turns out to be a complicated, genetically complicated thing, and in fact is a lot of the reason that biological control has worked in Europe and has not worked well in North America, because there is a lot of vegetative incompatibility among isolates here in North America, and much more vegetative compatibility among isolates in Europe. And I can talk about that more if folks are interested. So when you do that kind of experiment, of course, what you've done is you've moved everything that's in one fungus into the other fungus. And virologically, that doesn't tell you exactly what's going on. What you'd rather be able to do is actually purify virus out of the fungus so that you can actually find out what the virus is doing in the fungus, and isolate what's going on, why it is making the fungus sick, for example. And so to do that, you do the same things that other virologists do. You start with a virus-infected culture, and over a couple of days, you can purify the virus to a high state of purity. In this case, we can see this little band here is pure virus, and this particular one looks like that. And then you can take a fungus without virus in it, make so-called protoplasts, that is, make cells without cell walls from this uninfected fungus, and then mix the two together. Take the virus and take the fungal protoplast, mix them together, and plate that out. And then if you're lucky and you do it right, you can actually isolate part of that new colony that contains virus. So this isolate here, it's just another picture of this, of course, but this fungus then has exactly that virus. And so then you can fulfill what's called Koch's postulates and know that the effects that you're looking at are the effects of that particular virus on that fungus. We've done this with a lot of different viruses now. I'm not going to go into detail about these, but I do want to tell a couple of sort of quick stories about some of them. The first is this so-called hypovirus group, and it is the most important of the viruses that are associated with chestnut blight. It's the kind of virus that turned the fungus white in those pictures that I showed earlier. It's the virus that has succeeded throughout Europe to really do something about chestnut blight there, and it's the virus that it has been worked on by far the most of any of these chestnut blight viruses. It was in fact the first virus that was characterized, that we and others characterized, that had no protein coat around the virus. So those of you who are familiar with a virus like SARS-CoV-2, all of you should be by now, you'll think about the structure of that virus a little bit and know that it has a piece of RNA on the inside, and then it has a protein coat around it, and then it actually has a lipid envelope around it that, in the case of SARS-CoV-2, has spike proteins that are associated with that are encoded by the virus. So that's the typical type of virus structure, where you have a coat protein around the virus nucleic acid, and that coat protein protects it from the outside, and it also helps it get into the new cell when it sees a new cell. In the case of these viruses, there was actually no protein coat. All there was a lipid envelope around the genetic material, in this case, double-stranded RNA on the inside, and the replication-associated enzyme that let this RNA multiply. So this was a unique type of virus found at the time, because all viruses previously accepted by the International Committee for the Taxonomy of Viruses had a capsid protein around them. Why don't these? The bottom line answer about why these don't is because they don't have to, because that lifestyle that I talked about earlier, where the viruses tend to move from one fungus to another by fusion of hyphae, the viruses are actually never getting out into the environment. What they're doing is they're staying in the fungus, and they're simply moving across an anastomosis bridge from one fungus into the next. And so they never get out into the environment. And so they don't need that protection. And they also don't need any kind of receptor binding protein that allows them to get into the next cell. They just flow into the next cell. And so over time, they've essentially given up their capsid proteins because they don't need them. When we started working with these, we started to look at them genetically. I'm not going to go into this information for this particular talk, but what we found over time was that different types of the same family of viruses had different effects on the fungus. So the one that I mentioned earlier, this what I'll call CHV1, turns the fungus white. It's found in Asia and throughout Europe. It's again, very well studied virus. We found one here in New Jersey that was different enough from this one, and that causes a different phenotype or a different colony morphology in the fungus that we then designated it as CHV2. Found one in Michigan that is different enough that we called CHV3. And finally, one that was different enough that we designated CHV4. And this one, in fact, was found throughout North America. It is unique among these among the viruses in that it really doesn't do very much at all to the fungus. In many cases, you can't even tell that the virus is there. It's having very little effect on the biology of the fungus. So when we started out with that, looking at these viruses, we spent a long time thinking about how these viruses evolved from each other. How could CHV1 evolve to become CHV2 to CHV3 to CHV4, or did it go the other way around? Did it start with CHV1 that then acquired some genes to become more complex, et cetera? So again, you know, a fair amount of sort of thinking and hand-wringing about that, because these were the four known viruses of this type at the time. Fast forward to a few years later, as other people are characterizing more and more other fungal viruses through metagenomics research and things like that, we now know that these four viruses aren't each other's closest relatives. And in fact, CHV1 and 2 are closely related to each other, but they are not very closely related to CHV3 and 4. And in fact, CHV3 and 4 aren't even each other's closest relatives. So what that tells us is that these viruses almost undoubtedly came in separately, at least 3 separate times into this fungus. And probably the way it happened was by infection of from so-called sympatric fungi. In other words, fungi that are living in the same canker, for example, as the chestnut blight fungus is. And then an entry from one unknown fungus into this fungus. So what we still don't know, so now more than 200 members, so we start out with 4, and now more than 200 members of this virus family have been characterized. I'm not going to show a big tree that shows all that, but like I say, apparently 3 different entries of, at least 3 different entries of the virus into the fungus. Interestingly, CHV1 and 2 still, after those 200 members have been characterized, are still each other's closest relatives. So they may have co-evolved within Coinfections itself. We don't know that yet. Looking at the sort of worldwide distribution, looking at it on a bigger picture, something I'm interested in, and how these viruses have gotten around the world. We have very good evidence that the CHV1, that white, the virus that turns the fungus white, almost undoubtedly came from Asia to Europe. Again, that fungus is found all over Europe. Interestingly, CHV1 has never been found naturally in North America, so it did not enter North America naturally. CHV2, I mentioned we had found in New Jersey, and there's a nice community of that virus, of fungi infected with that virus here in New Jersey. It's been found once in China, and that finding is a little bit suspect for several reasons, but it's still entirely possible, and it makes sense that the CHV2 infected fungus would have come from Asia to North America. CHV3, which is only found in Michigan, there's no indication that it is in Asia or that it is in Europe. And in fact, its population suggests that it is unique, that it probably entered the chestnut blight fungus here in the Michigan area and radiated out from there, because it has not spread very widely. And finally, CHV4 is found all over North America. So it suggests that the entry to North America was early in the pandemic because it has spread so widely around. It doesn't look like spread that would have come about later on. What this leads us to hypothesize is that CHV4 infected isolates should be found somewhere in Asia, and so far those isolates have not been found. So again, we have evidence, the evidence suggesting that there's a long-term relationship of this virus, of this CHV4 virus, with the fungus, comes from a number of genetic sources, including the way the virus codes for proteins. I'm not going to get into that detail, but so what it tells us is that, and we're asking colleagues in Asia to, hey, start looking for this in Asia, see if it is there. So far, it has not been found.
Oh, Brad, we're about
Yes.
4 minutes. You got about 4
Okay.
minutes.
Okay, thanks. So I'm going to go through two quickie stories here. One is by looking at co-infections, by when one fungus is identified that has two viruses in it. Again, I won't go into detail here. This isolate that was found in West Virginia was found to have a different virus in it, but also one of the hypoviruses in it. And it turned out when we did the experiments, like I talked about a little bit before, that the this virus, the reovirus in the fungus was unstable. That is, the fungus booted it out easily. And when co-infected with the hypovirus, it then in fact stabilized it. And so the story turned out to be that the hypovirus, the CHV4 virus, suppressed the antivirus defense system of the fungus and allowed this other virus to enter and compete. So that has led us to think that that is probably happening a lot in this fungus, that a co-infection, sometimes you can get the viruses that compete with each other, but in this case, this virus is helping the other virus out by suppressing its antiviral defense mechanism. And so I'll finally finish with a quick word about these mitochondrial viruses that Jim Polishok characterized in my lab a long time ago. We started working with this one because it looked like an interesting little virus, and it turned out after a lot of work that the virus in fact resided in mitochondria, and that it had to reside in mitochondria, the so-called powerhouse of the cell. So it didn't get out of mitochondria into the cytoplasm of the cell. Again, an odd little virus at first. It didn't do much to the fungus. We worked on it a lot, but it was just hard to make a lot of progress on, other than the initial characterization and things like that. Similar to the hypovirus story, it has turned out that these mitochondrial viruses are all over the place. And so this one that was in Coinfections is one of them, but they're all over in fungi. At least 200 have now been characterized. And also they're now characterized in plant hosts. And we found a report of some in insects. And I talked to someone last week who's looking at them possibly in nematodes and C. elegans, which is a very interesting system to be investigating. And so I'm not going to talk about that last slide. So to summarize here, that these viruses, they're effective in biological control, but also they've been a very good avenue through which to look at virology in general. First viruses found to be devoid of capsid protein. First viruses found to be mitochondrial, to replicate in mitochondria. Reoviruses I didn't talk about here. But also, another thing I didn't talk about was the first attempt to actually deploy genetically engineered fungi and their viruses for biological control. And again, they've led to a lot of just interesting work on host pathogen interactions. So I'll stop there. I want to acknowledge folks. I'm not going to read all the names, but I've had a lot of great people associated with my program over time. And I note a couple of undergrads here, and my current graduate student, Alana, who's been a lot of help with the projects, and a lot of, again, collaborations, especially my former postdoc advisor, Don Ness, who I worked with a lot, and Nobuhiro Suzuki in Japan, where I did a sabbatical and brought some of this to him, and where we continue to collaborate. So with that, I'll stop and be happy to take questions.
Thanks, Brad. Just a reminder, if you do have questions for Dr. Hillman, you can enter them in the Q&A box at the bottom of the screen. We do have some right now. So you had mentioned science may take you in new directions from the one you started with. Is that something we need to communicate to young scientists, and how can we do that?
I think the answer to that is absolutely. The way I communicate it to young scientists, of course, just on an individual basis, on a one-to-one basis. When I have a graduate student, for example, well, whether it's an undergraduate or a graduate student, the thing that I will say to them is, early on, switch projects, switch labs. You don't want to work on whatever you're working on. You don't want to work on it forever. And if possible, switch coasts. Frankly, part of the reason that I came to New Jersey from California, I really wanted to work with the guy that I was working with, Don Ness, but I also wanted to switch coasts, find a different place. So those are suggestions that I make to people. And you also want to get involved with other systems just to keep your thinking fresh. So we started working on something that I didn't talk about at all today. We started working on transposons of this fungus about 15 years ago, and it's led to some really interesting work, something that I had just never thought about before. And so when you start to do a new project like that, you have to, a different part of your brain starts to think about it. You have to learn new stuff. So, I certainly say this in classroom situations. Don't get married to whatever you're working on too early on, if possible. Switch things around.
Okay, great. We have a question from someone who's currently retired. They ask, how can I get involved in the research or data collection in New Jersey?
I would say the American Chestnut Foundation is the easiest avenue. I will be glad to communicate with you about it, because again, we still do some projects that are New Jersey-based projects. I mentioned the one, the tree counting project that we did a few years ago with Rich Leopold. But again, I'll be glad to communicate with you. And again, through the American Chestnut Foundation, the person, New Jersey's chapter of that is actually associated with the Pennsylvania chapter currently. And so Sarah Fitzsimmons is the person who leads that chapter. So that would be a good place to start as well.
Okay, great. Another question, have we found similar viruses in other deadly plant-based or even animal-based fungi? That is, are there other fungal outbreaks that we could potentially combat with biological control?
Yeah, the short answer is yes. And that's why I pointed out in the case of the so-called hypoviruses and some of the ones that I didn't talk about that are found in other fungi. And there are a lot of them out there. So since I started in this area, this work has just come pouring out. One of the issues is that a lot of people just do the metagenomic stuff. In other words, they just grind up the fungus and look for viruses look for sequences of viruses basically, and don't really do the virology, don't really pull the systems apart well. But a good number of people are doing that, are doing very good virology. So with one of the important necrotrophic fungi called Sclerotinia, Sclerotiorum, and other Sclerotinia species, there are a number of these viruses that are around and that are being examined as potential biological control. There are similar viruses in Fusarium. There are similar viruses in an important root pathogen called Rosalinea. One of the main problems is getting transmission. So it's fine to have it to see these less virulent isolates, these hypovirulent isolates of something like Sclerotinia or Fusarium. The issue is, how do you get the viruses to transmit in nature? In other words, how do you make it so that you actually have an effective and cost-effective biological control? And so that's why it's the sort of important lesson about it working well in Europe because of the natural setting there, but not working well yet in North America is an important lesson.
Okay. Another question. Have you seen chestnut blight increase over the years of your study in our area?
The short answer is no, because we were wiped out. I mean, you have to understand, I guess I didn't make it clear enough, but that picture of the ghost forest that I saw, that I showed, that's what it looked like all over. So there was virtually no resistance. I mean, there were individual trees that people would pick out thinking that they were resistant, and many of them turned out to be just escape trees. So that fungus just came in. It was like dropping the fungus onto a petri dish of 4 billion trees, and it just ate through the 4 billion trees virtually without resistance at all. So the answer to whether I've seen chestnut blight increase, the answer is no. What we have seen increase is the size of recovering chestnuts. We don't know why that is. I suspect, like a lot of people suspect, that there is a little bit of blight resistance in those trees. And we know that there's hypovirulence. In the case of New Jersey, the CHV2 virus we can often find in recovering trees. So that's helping out. We also think that other fungi are helping out. So if the fungus, if Coinfections isn't killing trees right away, and other pathogenic fungi come along and start competing well with the chestnut blight fungus in the canker, then that's helping with biological control as well.
Okay. Another question: does human activity have any effect on the growth and spreading of the blight?
I don't think a lot. The human activity, again, and early on, attempts were made to stop the blight. Again, like I just said, it was unfettered. It just went through, and so there was no human activity that was increasing it because it just didn't need it. The main attempt to stop it, the greatest attempt to stop it, after people figured out early on by 1910, they had pretty much figured out that the fungicides would not work against it. The main attempt to stop it from spreading into the southern Appalachian range was in Pennsylvania, where a lot of trees were cut down. There was a major control program that was that the state legislature funded to cut down a lot of trees to try to make a break between where the epidemic was burning and the south. And people who knew chestnut blight, even at that time, thought that it was a fool's errand, and that in fact turned out to be the case. So a lot of trees were cut down in attempts to stop the blight, and that simply didn't work. And so again, in this particular case, there's been nothing, no human activity that has increased blight activity.
Okay. How did chestnuts come across the Bering Strait? Is it believed that they were carried by people?
I don't know the answer to that. I believe the answer to that is no, because it was way back when. But and I don't even know. I am the wrong person to be answering the question. I can tell you that. I don't even know that the Bering Strait theory is still the one that's thought to be the most likely. So I'll sort of take a pass on that. But the reason I don't think it was brought by humans is because it was thought to be so long ago. I mean, it was literally millions of years ago.
Okay. I think we have time for one more question, quick question. Given the value of chestnut trees to the economy, do you think that the blight ended up having a social impact beyond the loss of the wood, such as if chestnut trees had not died out, would the history of Appalachia be different?
Good question. Again, I'm kind of not the right person to answer it, but I guess I can answer it this way. I think it certainly would have been different, and it really, the epidemic, the pandemic, really did have a huge effect not only on the economy, but on the way people saw things, because again, groups of people who were very dependent on chestnut and who that was their tree community, suddenly you had this thing come through that just wiped it out, that just sort of changed everything. And so yeah, I imagine that it certainly changed people's mindset around it, and you could still, not so much now, but you can still talk to folks who remember when it came through. And for a lot of them, it was also at the time of the 1918 influenza pandemic as well. That was killing a lot of people. So I think a lot of people's mindset changed quite a bit. I remember my dad grew up in Mississippi, and I remember him talking about going off to college and coming back. So they had a lot of chinquapins where he was, and going off and coming back years later, and all the chinquapins were dead. So yeah, I think individually it really did have a big effect on a lot of people.
Okay, thanks, Brad. That's about all the time we have. I know that there were some questions we weren't able to answer. Please do know we'll be sending a follow-up email, and we'll include Dr. Hillman's email address as well as a video recording of this presentation. So thank you for joining us. Mary, do you have anything you want to say?
Just Brad, thank you so much. That was really fascinating, and I learned a lot about chestnuts, and I hope to learn a lot more just based on your talk today. I do want to let people know that our next and last Science Cafe is April 21st at 11:30. It's going to be a Thursday instead of a Tuesday, and Simi Payne will talk about High Seas Biodiversity, a Sustainable Future Through Science and Policy at the United Nations. So that's it for me, Brian, and Brad, thank you again.